Welcome back to our discussion about proteins. So here I have three different amino acids that I've drawn for you. So again, you notice all of them have an amino group and an acid carboxyl group. So that's what makes an amino acid. Here's three simple ones that we'll talk about often in the class. So this is called cytosine. This one is our simplest one because you notice the R group has just been replaced with a hydrogen. So this is our simplest amino acid. So you see it common. It helps provide structure because there's not much going on. And this will be an example of a nonpolar group. Here we have another one with a nonpolar group. This one is a fun one to say tryptophan. These one have these very interesting, actually, let me correct myself. This one might actually be polar because of the nitrogen. But tryptophan is another amino acid that has a couple of very interesting groups. You ever see this drawn different ways? They're all chemically equivalent. I draw the aromatics with these dashed lines. What that indicates is that the electrons are actually shared. So this is a little bit more accurate, chemically speaking, but you'll see other drawings. It's called tryptophan. Again, we have our amino group and our carboxyl group. And this last one is actually the most important. And it's most important, cysteine is the most important amino acid in a lot of ways because of this S. Again, we said some of these have sulfur, and let me be more specific. In humans, this is actually the only amino acid that has sulfur. So it ends up being a very, very common amino acid, one of the most common of the 20, because what can happen is this sulfur here is going to interact with sulfurs in other parts of the amino acid to allow folding. And when we go from just this list of amino acids, again, guanine, tryptophan, cytosine, instead of just seeing our protein as a lot of amino acids, which is how it's originally made, these amino acids are just floating around in your cell and they just get bonded together like we discussed in the last video. Your body will just literally take the oxygen here and chemically form a covalent bond between this oxygen, which has a negative charge in water, and it will bond to this nitrogen that has a partial positive charge. And through dehydration synthesis, we will release a water molecule. So we'll grab a hydrogen that's either floating around or from here, and an oxygen and a hydrogen from here, and they will form a covalent bond. So lose an oxygen, we'll get a covalent bond here, and this will happen in a ribosome, and will form these long chains. So we can see this as just a list of amino acids. That's called our primary structure. So if we were looking at just one of the amino acids, that's called a primary structure. So in this case, the primary structure of this segment of the protein would again be the guanine, tryptophan, cytosine. Next, we need to consider how do these different structures interact with each other, because as soon as it gets shipped out, it's not going to just stay in this linear structure, it's going to fold. And so because of protein folding, we go from a primary structure to a secondary, tertiary, sometimes even quaternary structure. So a secondary, tertiary, and sometimes quaternary structures. And we have a specific definition for each of these. And all of these structures have to do with 3D folding, have to do with the R groups of these amino acids. Now would be a good time to pause the video. Can you actually write out and identify what are the R groups? What's the difference when you say R group again? It's the difference between these amino acids. All of these have a nitrogen, so that's not part of the R group. All of them have a carboxylic acid. Sorry, I don't know why I can't say that today. Maybe I need more coffee. But what is different? What is the R group of each of these amino acids? Maybe you could email me what you think, or tell you if you get it right. But based off of the unique combination of the R groups, what we get is, again, emergent properties. We get very unique structures. And here's what you need to know about the structures right now. Secondary structures for this class usually come in two different forms. They're usually called alpha helixes, which I'll point out to you for in a second. And it's not working right now, which is unfortunate. So alpha helixes and beta sheets. These are the Greek letters alpha and beta. Tertiary structure, it just refers to the overall three-dimensional folding. So these are localized structures that are repeating patterns. Tertiary structure is the overall 3D structure, so that's easy to remember. The quaternary structure is when we have multiple polypeptide chains working together. Let me show you what that means by showing you the structure of myoglobin and hemoglobin. So quick shout out to Khan Academy. Love Khan Academy, great place for you to do some learning. So here we have Sal Khan talking about and showing to us the structure of hemoglobin. So first thing I want you to notice is all these like
because it's important, but also it explains a lot about how
Sorry, Khan. Need you to be quiet for a second. Okay, so the first thing you notice is all these twists. So all these twists here, these are all helices. So these are all alpha helices. The next thing is you might notice is some pleated sheets. Now, unfortunately, hemoglobin doesn't have a lot of these sheets. There's one right there. Yeah, it's just that happen to have more of the alpha helices than the beta sheets. Just will vary protein by protein, but all of these localized loops that look like curly hair, like my wife's curly hair, these are all called the alpha helices. They just have to do with the localized structure. But the overall 3D structure of a polypeptide chain is called the tertiary structure. Lastly, what I like about this picture from Sal Khan that he's explaining is it actually shows that there's two red groups and two blue groups. Actually, each of these were made independently. They're all a unique polypeptide chain with a unique structure. And they only are joined together at the very last, and that's called a quaternary structure. If we compare hemoglobin to myoglobin, which is a similar molecule, myoglobin actually only has one chain, so it doesn't have a quaternary structure. Still has a secondary structure, primary structure, tertiary structure, but hemoglobin is made out of 4 of these chains. So we call those interactions between multiple chains quaternary structure. And that's really all you need to know. So that was quick. That was my longest video so far, but it's very important. Everything you need, everything we're going to talk about is built off of an understanding of proteins are used in your cells, and that's what allows your cells to build you. So we're going to be talking a lot more about that, but that's it for today. Much love. Have a good day, everyone.